Hey, so today we're going to start talking about some of the things that we think of as really the main role of the kidneys. So we've talked a little bit about how they cooperate with the lungs to regulate the pH of your blood. But primarily when we think about kidneys, we're thinking about fluid balance and electrolyte balance. So we're going to be thinking about like the water content of your blood and the salt content of your blood. That's one of the main roles of the kidneys. Okay. So today we are going to begin talking about that main function of the urinary system. So the urinary system does do many other things. So we've spoken about the acid-base balance. So it's regulating our plasma pH. We're also going to be regulating the composition of that plasma in terms of what ions are there, what the osmolarity of the plasma is, so what kind of pull on water we have in the plasma, so that fluid surrounding your blood cells. Your urinary system is also working to remove metabolic waste products. So all those byproducts that we get as we break down molecules, we do want to eliminate them from your body. So that involves your kidneys. And we've seen in previous units that this is also a system that helps us regulate blood cells themselves. So your kidneys were where we were creating that hormone erythropoietin that triggered the production of new red blood cells. There are other kind of accessory functions along with that. So they produce something called renin. We'll get to that. They help us activate vitamin D. They do some gluconeogenesis, so some making new glucose that we talked about in A&P1. So they do a lot of things. But usually we're thinking about water, we're thinking about urine production, we're thinking about blood volume. Okay. So in order to do this, we are going to be working with basically a specialized capillary system. Okay, so we talked about capillaries before, and we talked about net filtration pressure before, NFP. So we're going to be modifying that now specifically to the kidneys as we think about glomerular filtration. So let's remember what our directions are. So what direction defines filtration? This is our direction word. And we had Starling's forces when we were talking about net filtration pressure. So we had forces that influenced fluid to move towards tissue. We had forces that moved fluid towards blood. We want to kind of tease out what those were as we now think about this specialized process going on in the kidneys. So start by picking your definition of filtration, and then I have your 4 Starling's forces for you as we saw them before with the NFP. They're going to be basically the same as what we're about to talk about now for kidneys. So we're going to pick 2 of them for our filtration direction. Take a look at how our recall is today. Okay, cool. All right. Yeah. So when we talked about filtration before and also now, when we're talking about filtration, this is a direction, and it's when we're moving fluid from the blood to a new location. So we're pulling fluid out of our capillaries for our filtration direction. So when we talked about net filtration pressure, we had 2 forces, 2 Starling's forces that propelled fluid in that direction. So we had our capillary hydrostatic pressure, So basically, like blood pressure in the capillary, push it out on the walls of the capillary. And as we push out on the walls of the capillary, some of that fluid moves through. So that's our filtration direction. We also had our interstitial fluid. It's oncotic or osmotic pressure. So that's a pull. So the interstitial fluid was lying next to the capillary. And when we had impermeable solutes dissolved there, so stuff that can't cross membranes, it pulls water towards itself. So that was our other force that moved fluid out of our bloodstream. That's basically the same process that's happening in the glomerulus as we are moving into our nephron. Okay. So at the top of our nephron, we have specialized capillary called the glomerulus. So it looks all twisty. It has an arteriole going into it. The afferent arteriole has an arteriole coming out of it, the efferent arteriole. That capillary, surrounded by the little Pac-Man called Bowman's capsule. It's going to be capturing the fluid that comes out of this capillary. Okay? So lots and lots of stuff is moving out of the glomerulus, is moving out of your bloodstream as it passes through your kidney. And it's actually about 125 milliliters per minute of fluid are going to be moving into the nephrons of the kidney, coming from your bloodstream, coming from those capillaries running past the nephrons. Okay. Along with that fluid, we're going to see many other things moving into the kidney. So into the nephron goes pretty much anything that's dissolved in the plasma, so long as it's not like protein sized. Proteins are a little bit too big to get filtered into a nephron, but things like glucose are going to be going, things like sodium are going to be going, medications may be going in, all sorts of stuff is coming into the nephron as the plasma is getting filtered from that capillary into the nephron. Okay. So in order to get from that glomerular capillary into Bowman's capsule, so into the beginning of the nephron, we do have to cross a couple barriers, right? So they're pretty easy to cross, which is why almost everything except proteins can do it, but we do want to know the pieces of it. Okay. So formally, the type of capillary that we have here as we're going towards our nephron loop, capillary is a fenestrated capillary. So a fenestra is a window. So these are like capillaries with big extra gaps in them. So it's like the opposite of the blood-brain barrier here, right? In your blood-brain barrier, you wanted your capillaries to be real tight, real sealed. We didn't want things moving from the blood into your brain. Now that we're in the kidney, we want to pass everything we can through the kidney so that we can figure out what to do with it, what to keep in the blood, and what to eliminate as waste. So those are our fenestrated capillaries. And you can see how we have these kind of slits here. Right, so those are our filtration slits here in our fenestrated capillaries. Okay, so we see these slits. We can see that we have podocytes on the other side here. So podocytes are foot processes, right? As we are catching the fluid kind of coming from the capillary, right? So if we're, let's say a glucose molecule or some fluid, I'll pick some water, right? So we're going to go through the fenestra, right? As we're getting filtered out, we'd cross a basement membrane. We're thinking of the basement membrane just as kind of gluing together two kind of cell layers. And then we're going to be going through these filtration slits that lie between these foot processes or podocytes here. Okay, so this is what's forming that kind of opposite side, this coating, this sort of like beigeish, orangish stuff that you can see coating the glomerulus, coating that capillary network here. Okay. Okay. So in terms of forces, we are going to see some variation in how strongly we're filtering stuff too. So 125 milliliters per minute is just kind of an average, kind of baseline. But exactly how much fluid is getting filtered into your urinary system per minute per day depends on these Starling forces, these pressures that we've spoken about before. Now we just have basically new locations where we're talking about them. Okay. So you'll see that, like when we were talking about our net filtration pressure, we got 4 of them, right? So we have 2 hydrostatic pressures, right? So hydrostatic pressures, we're thinking of as push. A hydrostatic pressure is a push. And our oncotic pressures, so the force exerted by proteins, is a pull, right? So hydrostatic pressures push fluid, oncotic pressures pull fluid. Okay. So then what we're going to do is think about, where are we pushing from? Where are we pulling to, as we figure out which of these are going to influence us to filter more fluid into our nephron versus which ones are going to oppose filtration, kind of limit how much fluid is going through the kidney. Okay. We can walk through those. So for our glomerular capillary hydrostatic pressure, right? So that's telling us the location is our capillary, the glomerular capillary, and it's a hydrostatic pressure, so a pressure coming from the pressure of fluid. So this is a push, okay? So we are pushing out of the glomerulus here. So that's this one, right? So we're pushing out of the glomerulus, out of that capillary, and into Bowman's space, which is that space in Bowman's capsule. So that kind of enclosed bit there at the beginning of the nephron. Okay, so that's a force for filtration, and we'll see it's actually a pretty strong one. Okay. Now, our other force for filtration is going to be coming from Bowman's capsule. So we have a new location, right? So from Bowman's capsule, the space is Bowman's space in Bowman's capsule, right? And it's an oncotic pressure, right? Our symbol for that is this pi symbol. So this one's a pull, right? Hydrostatic pressures are a push. Oncotic pressures are a pull. So we're pulling towards Bowman's capsule, which is why this is also a force for filtration. So you can see that arrow for our Bowman's capsule oncotic pressure is going the same way. So this is if you have anything dissolved here in this area, it would have a tendency to pull fluid towards itself. Okay. So we also have 2 forces that oppose filtration that could slow down this filtration process. So we're going to have a push and a pull for these forces in opposition as well. Okay, so we have a hydrostatic pressure that's in opposition. So any pressure that we have building up in Bowman's space, right? If you think about like a water balloon, it's harder and harder to put more stuff in there once it gets full, right? So this is going to be a push back towards the capillary from Bowman's capsule. So big P is our push. The subscript tells us the location. So from Bowman's capsule, we have a push back towards the capillary, opposing filtration. Okay. And we also have a pull that opposes filtration. So that pull is coming from the glomerular capillary system. So that's our location. Pi is for pull, our oncotic pressure here. So the proteins that we're leaving behind in the capillary as we filter out fluid are gonna a little bit say, wait, let's keep some of that fluid with us. That's going to be another force opposing filtration. Okay. So we're going to be using these forces to calculate or to think about the relationships that result in our total glomerular filtration pressure, which is the same thing as our net filtration pressure. We're just now being specific that we're talking about the glomerular capillaries specifically. So these capillaries in the kidney specifically. So we can see that that equation works the same as our net filtration pressure did, right? So here we can see we're starting out with our forces for filtration, right? So we start out by any arrows going this way, we add them up, right? And then we subtract away anything that's opposing that. So we subtract away those forces opposing filtration. All right, so here's our opposition. Okay, so any arrows that go that way are resisting that filtration pressure. So here we got explanation of some of our normal numbers for these here. So the glomerular capillary hydrostatic pressure, so that pressure of the blood in the glomerulus in your blood, in your blood system, your cardiovascular system, is usually about 60 millimeters of mercury. So that's pretty high. And it's higher than we saw when we were talking about the analogous process out in your systemic capillaries. And the reason for that is we basically have blood kind of backing up into our glomerular capillaries. So it seems like a little detail, but if you notice on these arterioles, right, we got a fat one and we got a skinny one, right? So we know that blood flows more easily with less resistance through tubes of larger radius, larger diameter, aka that fat one. Harder to get through those skinny little tubes because of the higher resistance, right? So because that skinnier tube is our efferent arteriole leaving the nephron, right? We get some backup of the blood, backup of pressure here in the glomerular capillaries. So that's why this pressure is higher than you see elsewhere in other capillaries. So our other force for filtration was our Bowman's capsule oncotic pressure. So as we're filtering fluid into Bowman's capsule, we should be leaving the proteins behind in the capillary. Like that's the one thing that should be too big, really, to get into the nephron, to cross through those fenestrae and then through those filtration slits in the podocytes. So this should be zero because we shouldn't have protein in our filtrate. If you do a urine analysis and you see protein in the urine, that's something you'd be kind of concerned about, actually. That is something they check. Okay. So those are two filtration pressures. And opposing this, we have our Bowman's capsule hydrostatic pressure. So that's the buildup of fluid in Bowman's capsule. So we filtered it into Bowman's capsule, and now it's exerting a little bit of a pressure in the opposite direction. It's kind of high too, not as high as our glomerular capillary hydrostatic pressure was, but we're filtering a lot of fluid pretty fast here. So there is kind of a buildup in Bowman's capsule as well. So that should be about 15 usually, because we have a pretty large volume of filtrate at the beginning of the nephron. We'll see how we adjust that as we move farther along. Okay. Now our final force is that glomerular oncotic pressure. So that's our proteins that are stuck here in the glomerulus, in those glomerular capillaries, exerting a pull on fluid back towards themselves. This is pretty high as well because we have filtered out a lot of the fluid as we're filtering out the plasma. And now those proteins are kind of just sitting there until they wash out through the efferent arteriole. So in total, that gets us to an equation that looks like this. Okay, so to find our typical glomerular filtration pressure, this is kind of our baseline expected textbook number. We're taking that hydrostatic pressure of the glomerular capillary and adding that pull from Bowman's capsule, which is usually zero. So we added together our hydrostatic pressure, the glomerular capillary, and our oncotic pressure of Bowman's capsule. And then we subtract away our opposing forces. So we have a little bit of opposition from the hydrostatic pressure that's building inside the capsule. And we have a fair bit more of a pull from proteins that are still in that capillary system. All right, so 60 minus 15, and we could distribute this negative, so also minus 29. right, is going to give us a total of 16 millimeters of mercury as our typical glomerular filtration pressure, which we will see abbreviated as GFP sometimes. Okay, so that's our typical glomerular filtration pressure, resulting in our kind of baseline amounts of filtration and therefore baseline amounts of urine production. Okay. So this is related to, but not exactly the same as the glomerular filtration rate. So the reason I wrote GFP on the previous slide was partially to highlight for you that you want to pay attention to what the last letter is here, right? You want to know whether you're thinking about the pressure, right? So the force for filtration, or whether you're thinking about a rate, whether you're thinking about the actual like milliliters per minute or liters per day of fluid moving through the kidneys. Okay. So when we're thinking about the glomerular filtration rate, we are thinking about how much fluid is going into the nephron, and we got a time on that. That's why it's a rate. Right. So to look at this, we're going to think first about how much blood is flowing through the kidney just in general, right? So that's what our renal plasma flow refers to, is how much blood went to your kidney per minute. And it's a lot. Lots of blood goes to your kidney. On the order of 625 milliliters per minute of blood is going to your kidney as it circles around through your cardiovascular system, gets mixed up in the heart, and moves out again to all the organs. Okay, so here we can see that plasma, which is just the fluid part of our blood, coming in through the afferent arteriole. This network is our glomerular capillary, and we can see that we have plasma also leaving the glomerular capillary, going back to your heart, going to get oxygenated. We don't take all the plasma and put it into the nephron immediately. So we can see that we still have some plasma leaving, right? So there's still some plasma leaving our kidney. Okay. So for our glomerular filtration rate, it's going to be the difference between those two numbers, right? So if we had 625 liters of fluid in the plasma coming into the kidney and you only see 500 milliliters leaving, right? That 125 went somewhere, and where it went was into the many nephrons that compose your kidney. So that's what our glomerular filtration rate is how much of that fluid is going into the nephrons, into the kidney to get further processed to determine how much of that fluid you actually want to keep in the blood. We'll do some adjustment of that later on in the nephron, right? Not right here at the top of the nephron, but in the later portions of the nephron, the more distal portions. Okay. So compared to systemic capillaries, this rate is high, right? So our filtration pressure in a systemic capillary, when we looked at the NFP, is about 2 millimeters of mercury. We just saw that our glomerular filtration pressure, which is the analogous number, is 16 milliliters of mercury, right? So the filtration rate is correspondingly higher as well when we think about the kidneys, right? So for our filtration rate for our systemic capillaries, we were doing like 3 liters per day. If you multiply that glomerular filtration rate of 125 milliliters per minute out, you get to 180 liters per day of fluid are moving into the nephrons in your kidney, right? So you're absolutely not going to pee all that fluid out. That's not all going to get excreted at the end. Should immediately emphasize to us that we're going to have to adjust this because you definitely do not have that much fluid in your plasma. That's what the rate is. We can also think about this relationship as a fraction, right? So the filtration fraction is basically just like, what percent of the fluid did you actually put into the nephron? What percent of the fluid is in the kidney getting processed? So for that, you're going to take your glomerular filtration rate, right? So that's that 125 we just looked at. And you're just going to divide it by the renal plasma flow, which is just a fancy way to say how much blood went to the kidney in the first place, right? So that's our 625 milliliters per minute. Right? And that's it. We literally just take a percentage. So we divide those two numbers. You get a decimal 0.2 that you can then convert to a percentage. So we have our filtration fraction of 20%. So 20% of the blood that's going through the kidneys every minute, every day, is going to, at some point, end up in the nephron, getting processed, getting adjusted, which is good because that plasma had all sorts of things dissolved in it. We may want to eliminate those out in the urine. We may want to pull them back into the bloodstream. Your kidney is helping determine that. Okay. So when we think about glomerular filtration and we're focused on what's dissolved in the plasma rather than just the fluid itself, we may refer to the filtered load. Okay. So the plasma is the fluid in the capillary. Right? It's moving in large quantities into the beginning of the nephron, and it's taking everything but like proteins and cells along with it. So particularly, we'll be interested in things like glucose is a common one to test and to get checked. So anything that's small enough to filter through those fenestrae and then through our filtration slits is going to end up in the nephron. And it's going to end up in the nephron in basically the same amount that you saw it in the plasma. So all we do for filtered load is we are going to multiply our rate of filtration, right? So how much fluid did we filter into the nephron, the beginning of the nephron? We're going to multiply that times that concentration of whatever molecule we're interested in as we might measure it in the blood, right? So if you're looking at glucose, you'd multiply glomerular filtration rate times the concentration of the glucose in your blood plasma. If you're looking at some specific medication, just be glomerular filtration rate times the concentration of that medication. If you're doing salts, glomerular filtration rate times the quantity of salt in your blood plasma. Hopefully pretty straightforward. So that's what our filtered load is. Okay. So this is going to depend on how much of that molecule you have in the plasma, as well as on the glomerular filtration rate, right? So this is going to go up if you increase glomerular filtration rate, right? If you pull more fluid, more plasma, essentially, into a nephron, you're going to be pulling more of whatever that molecule of interest is. So glucose is a particular one that we will track. The ways we test for diabetes is we analyze the presence or absence of glucose in urine, right? So glucose is freely filtered into the nephron along with all that fluid from the plasma, right? So it goes in exactly the same concentration as we saw it in the bloodstream. And that concentration happens to be about 100 milligrams per deciliter, which we could convert to get 1 milligram per milliliter. We would do that conversion in order to match what we have as our units here on our glomerular filtration rate, right? So glomerular filtration rate, 125 milliliters per minute. All right, times our concentration of glucose in the plasma. We're going to get 1 milligram per milliliter. I'm not going to be testing you on conversions in a lecture, so yeah, I'll give it to you in the right units if you have to do this. So we can see here that we can eliminate our units here. So we're going to get 125 again, because we're just multiplying across, leaving our units here as milligrams per minute, okay? So now we know our filtered load for glucose, right? So we have the amount of glucose that is passing into the kidney, passing into the nephrons every minute. Okay. Now, it is important that we regulate our glomerular filtration rate, right? So glomerular filtration rate, 180 liters of fluid being filtered every day is an awful lot, right? When we see changes to that rate, we're going to see changes to filtered load of anything that is dissolved in the plasma and coming along with that fluid. So we want to keep this within a kind of reasonable range, okay? Now, part of this that we're going to notice from that 180 liters of fluid filtered per day already is not how much fluid you are urinating out per day. So we do have more adjustment as we move through the nephron of how much of that fluid actually stays in the nephron, in the kidney, and makes it to your bladder. You should only be urinating about 1.5 liters of urine per day. So excretion is the direction physically out of your body, right? So most of that fluid is getting reabsorbed. Same thing for many of the substances that we have dissolved in the plasma and present in the filtrate. We're going to see a lot of reabsorption. So reabsorption, reminder of that direction. All right, so here's our nephron. Okay. Here's our bloodstream, afferent arterial coming in. Here's our glomerulus, that glomerular capillary network, efferent arterial going out. The blood continues to kind of neighbor the nephron as we go through. This is not to scale, right? But we have our filtration direction is fluid moving into the nephron. Our reabsorption direction is going to be fluid and other molecules moving back to the blood later on. So when we see small increases in glomerular filtration rate, you see a lot more fluid filtered, right? Which means the filtered load of things like glucose goes up, which could potentially result in more excretion unless we change that reabsorption further on down the line. Okay. So for this reason, we're going to regulate our glomerular filtration rate to try and avoid big problems here. Okay. Glomerular filtration rate is happening because we have a glomerular filtration pressure created by those forces for filtration, the main one of which was that hydrostatic pressure of the glomerular capillary, right? So the force of blood pushing out on the walls of the glomerulus was our big force that's forcing fluid into the nephron, okay? And the glomerular capillaries are capillaries, so they will be influenced by your blood pressure, right? So there's a relationship, certainly, between our glomerular filtration pressure and our mean arterial pressure, which means that there's also a relationship between our glomerular filtration rate and our mean arterial pressure, because if your filtration pressure goes up, that's going to mean that the amount of fluid going into the nephron is also going up, okay? So here we see the relationship between mean arterial pressure and our glomerular filtration rate. Okay. So we can see that in general, the relationship is that if our mean arterial pressure goes up, our glomerular filtration pressure goes up. right? So as we increase mean arterial pressure, we increase our glomerular capillary hydrostatic pressure that push into the nephron, which means filtration is going up. But we can see that this isn't a one-to-one relationship as well, right? So we can see that we've got this flat range in the middle. We've got this kind of plateau. So that's our regulated range. So when I say that we're regulating our glomerular filtration rate, this is the kind of normal range of mean arterial pressures, our normal range of blood pressures that we are equipped to handle through the mechanisms we're about to talk about. And you can see if we get outside of this range, right? So if your blood pressure really drops low or really shoots up high, our regulation isn't doing so hot, right? We start to see way bigger effects on the glomerular filtration rate if we get outside of this range. Okay. So when we're regulating our glomerular filtration rate, we're going to be thinking about three forms of intrinsic regulation. So intrinsic, we're thinking local, right? So local to the kidney here. Okay. The first of these is myogenic regulation. So this is about the smooth muscle, okay? So like our other blood vessels, those arterioles that go into the glomerular capillaries have smooth muscle in their walls because they can change their radius, right? So they can change their resistance by changing their radius, changing how much is flowing into them. Okay. Now, the smooth muscle in these arterioles has an automatic reaction in response to stretch. Right? So if we imagine we have a lot of arterial pressure coming in, right? So your blood pressure is spiking, mean arterial pressure is up. We're opening kind of a fire hose generally through our cardiovascular system. It's coming to the kidneys. Your afferent arterioles, the ones that go to the nephron, to the glomerular capillary, are going to stretch as we have that force of fluid kind of coming in. In myogenic regulation, that smooth muscle in the wall of the afferent arteriole doesn't like that. It bounces back. So what we're looking at when we're thinking about that myogenic regulation, right, is we're noticing here. So here's our afferent glomerular arteriole, right? So the blood flow in, right? So we're thinking about the effect of those walls kind of bouncing back. So smooth muscle in the wall would have a tendency to bounce back when it's stretched out, which means that we're going to kind of slow down the flow of fluid going into the glomerular capillary. We're going to keep that glomerular capillary hydrostatic pressure under control, even though we otherwise have a high pressure throughout our body and kind of faster, greater flow moving all around. So that's one way that we're regulating our glomerular filtration rate. And in essence, trying to keep it down is what we're focusing on there. Okay. Our next piece is tubuloglomerular feedback. So when we're thinking about this, tubulo is the tubules of the nephron, right? So tubules here. So you can see, we kept the color kind of consistent. So here we can see parts of the nephron are coming up here. So we have some distal parts of the nephron that are shooting back up towards the beginning of the nephron, right? So if we look in order, right, we're thinking about this part right here. Those kind of end parts of the nephron coming back to pass the initial blood flow going into the glomerulus. That's the glomerular part. So tubuloglomerular, so tubules that are passing our glomerulus. I'm not going to rewrite glomerulus. It's in the word, right? So we have feedback coming from the tubules to the glomerulus that are telling us something about the flow of fluid passing them, right? So they're basically able to sense, oh, hey, we suddenly have a lot of fluid, a lot of filtrate passing. So we're going to be potentially excreting, urinating out a lot of fluid, Do we necessarily want to do that, right? So that's what's happening with our tubuloglomerular feedback. So the mechanism for this is that we have some cells helping us out. Chalking. So we're thinking about our macula densa cells here. So our macula densa cells, this color's not working too well. Okay, our macula densa cells are here in the walls of our ascending limb of the nephron loop. So those distal parts of the nephron, those parts that are filled with our mostly or at least partially modified urine. Okay, so macula densa cells. And they're going to be able to emit paracrine factors, which are just local signals. Endocrine goes to like a different organ system. Paracrine just kind of goes to local neighbors. So those macula densa cells specifically, right, are communicating with these granular cells that are coding our arterioles going into the glomerulus, right? So they're sending us a signal telling the afferent arteriole how much fluid is about to exit the nephron. And the smooth muscle of the arteriole can respond to that. Particularly if you have lots of fluid that's about to leave the nephron at the end, that might be a sign that you want to contract down that afferent arteriole, because that's probably a sign that your glomerular filtration rate is getting high. That's why we have this fluid rushing past. So this is another way that we'd be keeping that glomerular filtration rate in check. We'd clamp down, increase resistance. So we're lowering flow, slowing down the blood going into the glomerular capillary. The final piece of our intrinsic regulation is mesangial cell contraction. Okay. So I want you to notice those purple cells, the macula densa cells are not all of them, right? So the macula densa cells here were the ones that are right here as part of the tubule, as part of this ascending nephron loop. Mesangial cells are the ones we have kind of here, but you can see them all throughout, right? So all the rest of this purple stuff kind of. Okay. So mesangial cells are in there, kind of jumbled in with the glomerular capillary, right? So the glomerular capillary made of an endothelial cell layer, and then those podocytes that are kind of lining those capillaries. Okay. So what happens with the mesangial cells, right? They're also kind of reacting to what's happening as we see glomerular filtration rate spiking. If we see it going up, they can contract. And since they're all in here, kind of coding and interspersed between those little loops of the glomerular capillaries, they can basically contract down and kind of turn off parts of that glomerular capillary network. And if they do that, we've essentially made it harder for fluid to get through those walls of the glomerular capillaries and move into the nephron, right? So we're slowing down the filtration by basically giving ourselves less surface area to work with. You can also kind of think of them as, it's not exactly what they're doing, but you can think of them as kind of covering over some of the pores or some of those filtration slits or something, or making the filtration slits a little smaller. Okay. So all three of these are in particular ways you might be dealing with spikes in mean arterial pressure. Could go the other way as well, but mostly this is something we think about when we think about like high blood pressure and how we adjust for that, right, as we see rises. And certainly in the direction we've written all these words, this is assuming that your pressure went up and you're trying to pull it back down. Okay. Which matches up with this flowchart. Okay. So what we see happening, what we see happening in this flowchart, right, is we're thinking about, okay, how do we deal with a mean arterial pressure that has gone up, right? So our mean arterial pressure goes up, which means the pressure in the arteriole goes up, right? So the pressure in the arteriole going up means that absent any regulation, the glomerular capillary pressure is going to go up. If the glomerular capillary hydrostatic pressure goes up, That means filtration pressure is going to go up, which means filtration rate is going to go up. All right. Now, we don't want that to get out of control. We want to keep ourselves in our regulated range, a range that's good for filtering out efficiently the molecules that we're trying to filter out and reabsorbing the ones we want to reabsorb. So to regulate that, we've got our mechanisms, right? So as that afferent arterial pressure is going up, that's stretching the arterioles. They bounce back. So that immediate myogenic response here is constriction. Now our resistance in that blood vessel going to the glomerulus is up, which means that we're pulling down that glomerular capillary hydrostatic pressure. So we're basically turning off this effect here, right? So we're trying to turn off that rise in glomerular capillary hydrostatic pressure particularly. Now we can see our other pieces of the regulation here. So here we can see that another thing that's happening, right, is if you let this flow all the way through, right, that's the same thing that we have here, right? So if we're dealing with a high glomerular filtration rate, those macula densa cells in the distal parts of the nephron are sensing a greater flow of fluid past them. So they're sending out their paracrine signals, so those local signaling molecules. And the reaction of the afferent arteriole is to constrict, which increases the resistance, which is another way we're doing negative feedback, turning off that increase in glomerular capillary pressure. So these are two of the three ways that we intrinsically regulate the glomerular filtration rate. Okay. There is some extrinsic control as well. Because there's intrinsic control of blood pressure that we talked about with the cardiovascular system. So we're not going to focus on the intrinsic control here with the kidneys, but we remember that we have, for example, the baroreceptor reflex as you are seeing drops in blood pressure, right? Okay, so we can see those same extrinsic controls having an effect on our glomerular filtration as well. Okay. So those three factors are mostly dealing with like, if we see glomerular filtration rate go up, when we're thinking about glomerular filtration going down, we might be thinking about some of those extrinsic controls that have to do with regulating your blood pressure more generally. Okay. So I'll let you go through this flowchart on your own if you want. We're not going to jump back into the details of that much in this unit. So when we talk next time, we're going to be thinking about reabsorption. So go ahead and review that at some point over the weekend. We're going to be doing some calculations about particularly transport maxima and renal thresholds is the thing that would be helpful to kind of preview before we talk about it.